Well, good evening to everyone who has joined us. Welcome to the webinar. My name is Ed Hoyt. I have the honor of serving as President and CEO of Calroy Health Sciences, and our company is presenting this webinar tonight. Our mission is to promote new science-based solutions to major health challenges that will improve the lives of millions of people worldwide. Tonight's seminar is about the role of the endothelial glycocalyx, a structure that covers the endothelial layer throughout the body and which regulates almost all endothelial function. Dr. Corey Rice, tonight's speaker, will be sharing the presentation with us. At the tail end of it, he'll be talking a little bit about some of the questions we've been getting asked from some of our prescribing practitioners about the endothelial glycocalyx role as relates to COVID-19. And then we'll also open up to questions and answers. There is a question and answer dialog box on the for most of you on the right-hand side of your screen where you can put in your questions. If there are questions that we can answer during the seminar, they will be, but more technical ones we will take at the end for Dr. Rice or Calroy's co-founder, Dr. Chen, as our Chief Scientific Officer, will be able to also help answer some of those questions. So I'd like to introduce Dr. Rice now and have him take over. He, by the way, will be presenting for about 30 or 35 minutes, and then we'll have Q&A. So Dr. Rice graduated from Baylor University with a bachelor's degree in forensic science and then completed his medical school at the Arizona College of Osteopathic Medicine in Glendale, Arizona. He attended Methodist Medical Center of Dallas for his internship and residency in internal medicine. He was chosen as chief resident of his internal medicine residency program during his final year. He's gone on to create a very successful practice. He has a concierge practice, and he will share with you as he's going through his seminar some of the dynamics of his practice and some of his clinical experience as it relates to tonight's subject. So I'm happy to introduce Dr. Corey Rice, and I'll turn this over to you.
Excellent. Thank you, Ed, and thank you everyone who's on this webinar tonight. I don't think we've ever had a time right now where time's more valuable than it is today. So the fact that you are spending your time with us means the world to us, and I hope that we can make it worth your while. Before I get going with a pretty amazing concept that I've been immersing myself in, just a brief statement about COVID. I know we're all in uncharted territory right now as clinicians that are seeing patients. I hope a lot of you that are on this call are still able to see patients, and I think our thoughts and our prayers are certainly going out to those who are affected, but also I'd be remiss if I didn't discuss the first line responders who are out there, treating these patients in our ICUs and our hospitals. My background is actually fairly conventional. Before I transitioned over into functional medicine, and I was a hospitalist intensivist working in the critical care units. And so, doing IV fluid resuscitation, pressors, and vent management was sort of my life. And so I empathize with those who are having to deal with that now. And I hope that countrywide, nationwide, we start to prioritize these physicians and these nurses, because I don't think the layperson understands that we don't really have a deep bench of qualified, competent healthcare providers to just jump in at a moment's notice to help run these ICUs and these critical care units. So my thought is certainly with them tonight, but that's a great dovetail, I think, into tonight's conversation, because we're going to be talking about a structure that is gaining momentum in the literature and how that particular structure, relates to cardiovascular health, cerebrovascular health, and really systemic health. And then we'll get into how we could potentially repair that structure and what potential pathologic conditions that we see day to day, day in and day out, that can be affected by treating that particular structure. So the glycocalyx in and of itself is something that we're going to be talking a lot about, and I encourage any listener to do your own lit searches and PubMed searches on this particular structure, because you're going to be blown away at the amount of literature that's out there. So looking at our health overall, 60,000 miles of our human body is blood vessels, and just to put that in context, that's 21 and a half round trips from LA to New York. So you lay your blood vessel out and you go to LA from New York and you do that 21.5 times, that's how far that is. So we're talking about a long stretched out blood vessel here. So to say it's the foundation of our health is not really a stretch. Controls several different functions in and out of the body, certainly detoxification, oxygenation, transport of hormones, nutrient exchange, immune function, vasoactive substances like nitric oxide, and helps to regulate really the health and function of every organ. And we're referring not to just the vascular system, but the microvascular system as well. So tonight we're going to focus a little on the endothelium and then get into this sort of finger-like projection here, the glycocalyx. But the endothelium itself, it's a single layer of cells. A lot of this, remember this out from medical school and residency, but the single layer of cells that we once thought was really our first line of defense before we compromised the vessel wall. But the role of this endothelium is to maintain vascular tone, healthy blood pressure. It's really a sieve to help, things coming in and going out, and really the permeability of vascular transport. It helps possesses antithrombotic and fibrolytic properties, modulates interactions between the blood vessel and circulating leukocytes and some of these adhering platelets. And then again, it helps secrete nitric oxide that ultimately helps regulate various functions. And again, my focus is going to be really on the outside here. So I love this depiction because it's a cadaver artery cross section. And the vascular lumen, to those listening, this probably reverberates with you a little bit. It certainly does to me. I do a fair amount of high-end cardiovascular risk stratification in our locations of our practices, and we're looking at everything from particle sizes to oxidated LDL to inflammatory markers and all of this stuff to get a risk profile of someone's overall cardiovascular health. And so the focus has always been really right there in the lumen and what's circulating. Is it toxins? Is it atherogenic particles? Is it oxidated particles? What is it? And there hasn't really been, a lot of focus here in the vascular wall. And so what we're kind of reorienting our brains to, or at least accessing our brains to tonight, is the idea of what's actually happening on that layer of the endothelium, and then the structure that then protects it, which is the glycocalyx. There really is no medical specialty that focuses on that specifically. So it doesn't matter who you are on this call and what you see on this call. I can almost guarantee you there's not a patient you see, treat, or otherwise operate on that wouldn't benefit from at least having an awareness of that layer, because I'll show you tonight that it's really that layer that might be a little bit more of an important focus than just what's circulating in the lumen. I think there's this paradigm shift in cardiovascular medicine to look more at that endothelial surface and things that regulate permeability in and out versus just what's circulating inside that lumen. So keep this cross section in your brain as we go through this. Now, just a little pathophysiology of arterial breakdown. A lot of you already understand this. There's a very well-known triad of vascular compromise or vessel compromise, one being inflammation, one being oxidative stress, the other one being endothelial dysfunction or dysregulation. So ultimately what you have is this sort of fire that starts brewing. It weakens that endothelium that I was showing you with one single layer of cells. The arterial wall starts to thicken. You then have lipids and these atherogenic particles that start to build within the wall of the artery, leading to atherosclerosis. And then you get these sticky sort of adherent platelets and leukocytes that start to adhere to that endothelium. And really that first step in atherosclerosis is that leukocyte adhesion. Then you sort of have this picture here. This is another pretty cool cross section. You've got this powder keg dynamite that's on the other side of the wall of the lumen, that's just sitting and it's just waiting to explode and go off. And when that happens, you end up getting a clot that forms right inside the vascular lumen. And what becomes important is, where is this occurring, right? If it's in the coronary vessels, whether they're small, large, or what have you, you're going to have an infarction. If it's embolic and it's in the cerebrovascular system, you're going to have a stroke. This could be all the way down to erectile dysfunction and peripheral artery disease. So the kind of clot we're referring to, by the way, this isn't typically the calcified stenotic clot or plaque. We're talking about the thin fibrous capped, lipid-rich necrotic core, the vulnerable rupturable plaque that's just waiting to explode. Vulnerable rupturable plaque is really the overriding cause of coronary artery disease in this country, is really from that type of plaque. Calcified plaque isn't really a problem unless it becomes stenotic. So it's really talking about this sort of nasty material here, is really what we're trying to prevent, and again, shift our paradigm to looking at the other side of the wall than just what's inside of the lumen. One of the issues is these diagnostics we have are pretty advanced and pretty revolutionary, but as a lot of you on this webinar can attest to, oftentimes it doesn't get to our clinic, our brain, or our treatment armamentarium or our diagnostic armamentarium until 10, 15 years after the technology was actually discovered. And so what that's leading to is, we have the capability to detect some of this plaque or some of this inflammation or oxidative stress early, but the focus, at least in outpatient and then conversely into inpatient management, is really the end stage of that disease. And so globally, we're certainly seeing mortality rates go down, but the prevalence rates of these conditions and really the cost just continue to go up. So it's the tools are there, and I think the diagnostics are there. It's just slow going in getting those implemented routinely across the board, but then also having interventions that can produce palpable, sustainable clinical outcomes. So it really sets a stage for a new approach and looking at something else other than just what's circulating. What can I do to lower what's circulating in the vascular lumen? And I don't want to say that's not important. It certainly is. But if we can emphasize and look more about what's happening in that single layer and how do we prevent things from penetrating through that wall to create that powder keg dynamite image I just showed you, that's really where I think the focus needs to shift, and I can already tell you that it is. And so really we're focusing more on where is the root cause of this, right? How do we prevent this stuff? It's not just about what's in that lumen, but how do we prevent anything from actually penetrating through to that single layer? So looking back historically, we have to learn from history to advance future, right? So looking back historically, when you look at this image was from a long time ago. But back then, let's say 10, 20, 30 years ago, probably more like 30 years ago, when you look at this image of the vessel, you're dealing with a very, very thin structure here. And it wasn't a stretch to say, I would conclude by that image that the vascular lumen was likely touching the endothelium. It looks like it's communicating pretty nicely. And so that was really the pervasive thought. Then we started getting better imaging capability and technology. And what we were able to look at was this invisible barrier here. And you can see where the arrows are. You actually see the lumen not touching the endothelium. At the time, we didn't know what that was or why that was happening. The pervasive theory was that it was some form of electrical charge that was kind of separating the endothelial layer from the lumen itself, but there really wasn't a clear idea of that. Then we got more sophisticated imaging, and we were able to actually go inside a vessel, and there it is. We noticed now what we know to be is the endothelial glycocalyx. So snapshot, just like you need to have that cadaver artery snapshot in your head with that powder keg plaque about to explode into the vessel, you need to have this snapshotted in your head as well. This little hairy-like, finger-like projection, you can think of this as however you like. You can look at it as a furry coat that sort of surrounds that endothelium. This is a very, very important structure, and it doesn't matter if we're talking about small vessels, medium vessels, or large vessels. This is head to toe, 60,000 miles of blood vessels is lined by that particular furry coat. And it's that furry coat that houses and keeps all sorts of enzymes and proteins and all of these things that regulate really overall vascular health and endothelial health. This is actually a sugar coat, so it's very, very carbohydrate rich. It's a very slippery membrane that's gel-like. It's made of these glycoproteins, these proteoglycans, glycosaminoglycans, all of these things that really make up the health of the endothelium. But the key for you to know is that particular structure there, and I'll show you some things as we move forward, is easily degraded but also we've discovered that it's easy to also regenerate that structure. So we're not just talking about the coronary vessels, the carotid vessels, we're talking about systemically head to toe vessels that have that particular structure on it. And that's again, the glycocalyx. So what has research done? This was actually theorized all the way back in 1917, but we didn't visualize this on some form of imaging until about the 70s. And once we were able to see a hazy image of this glycocalyx, or this furry sort of membrane or furry coat, the research exploded. And as you can see, as we got into the 2000s and 2010s, there's just a lot of articles out there on the glycocalyx. Certainly, as I said, if you do your own literature search as of right now, you'll find a lot out there on the glycocalyx and ways to help it and different conditions it's involved in. So what is the role of the glycocalyx? I mean, this is where the rubber meets the road. Like, what does it actually do? What does it actually control? And so there's a lot here, and I could spend a lot of time on this slide, but I'll kind of hit this fairly quickly for time's sake. But as I said, it acts as a sieve. So it regulates things going in, going out. This could be things like small dense LDL, oxidized LDL, Lp(a), you name it, activated platelets, sticky leukocytes. All of these things are being regulated by the glycocalyx to not penetrate that endothelium and then lead to that powder keg sort of plaque that I was referring to earlier. It ultimately helps to regulate the inflammatory response. It's very slippery, as I said, so it's an anti-adhesive. It helps harbor coagulation regulatory factors, clotting and anti-clotting factors. And then very importantly, it houses the superoxide dismutase. So this is massively important. This is an enzyme that catalyzes and breaks down superoxide anion, all of which is a big role in oxidative stress and helping lower that. As a lot of you probably know, oxidative stress, the idea of that is the arteries or the vessel is rusting from the inside out. It's like an old home with old pipes. Sometimes you have to replace those pipes because those pipes oxidize or they rust from the inside out. Exact same thing happens to arteries, exact same thing. So this particular sort of internal pharmacy or enzyme itself helps to prevent that rusting of our pipes, so to speak. It also triggers endothelial cells to produce nitric oxide, like I mentioned. Nitric oxide, if you have a compromised ability to make nitric oxide, which again happens in the glycocalyx, you could lead to all sorts of issues, obviously blood pressure problems, erection problems, you name it. So it's multifold on the role of this structure, but it's very dynamic. It's changing, it's connected, harbored and anchored into the endothelium, and at all times it's sort of this dynamic moving structure. So the glycocalyx, it is a proxy for endothelial health. So my hope is that as you move forward thinking about this structure, you understand that you can't have a healthy endothelium if you don't have a healthy glycocalyx, and you can't have a healthy glycocalyx without a healthy endothelium. So these two things have a sort of, symphony dance together where one can't function without the other. So they really do work together in unison very nicely. Now, what causes the degradation or the breakdown of the glycocalyx? This is important because this is the stuff you and I and the clinicians on the call see all the time. High levels of glucose, high levels of insulin. So specifically 85 milligrams per deciliter. If you've got a blood sugar above 85, it potentially is destroying the glycocalyx. A lot of you check beta cell function like I do, and you're following fasting insulin, right? That's the first sign that you're getting diabetes. Once the beta cells break down, that in and of itself is very toxic to the glycocalyx and absolutely will degrade it. Inflammation, right? It's kind of an all-encompassing term, but inflammation absolutely destroys the glycocalyx. I just talked about the rusting of the inside of the pipe, so oxidative stress certainly does. Toxins, who isn't exposed to toxins, right? That vascular lumen that has all of that toxic material, whether it's stored in fat, whether it's something they eat, whether it's something they touch, breathe, or drink, it doesn't matter. Those toxins circulating through that vascular lumen tend to attach to that glycocalyx and start to erode it or degrade it. So that's the whole idea of detoxification. And I guarantee you some people on the call here have a big supreme sort of respect for detoxification and helping people do that better. Certain infections cause it, lifestyle factors, uncontrolled stress, sleep that isn't controlled, or sleep apnea that isn't controlled is also it. Certain genetic factors and aging. I'd also add to this list, it's not on here, but hypervolemia. So anyone that manages fluids, if you're a nephrologist, if you're an intensivist, whatever have you, that you're actually doing fluid management, there's a huge sort of movement of research around sepsis, septic shock, but also just hypervolemia, and someone that goes into the ICU that's critical. And as someone who used to not only work in those areas, but study in those areas, I can tell you that fluids were kind of our lifeline back then. We would just flood people and hope their kidneys wouldn't give out. Well, now we're starting to learn that the glycocalyx in and of itself, too much fluid is not good on the glycocalyx, and that sort of parlays into sodium as well. So I would actually add here too much fluid, so too much volume expansion and too much sodium can be somewhat toxic to the glycocalyx. But as you can see, I mean, all of these processes sort of age and weather our arteries, but at the end of the day, they're also aging and weathering the glycocalyx. So you need to start thinking about vessel health, arterial health, the same that you would as really just the glycocalyx health. They're all sort of in the same paradigm. All right, I love this picture because this is hitting home with all of us who practice medicine on the call. So these are all the things we see and we treat every day. And if you could have one treatment, or I know we don't practice medicine in a vacuum. However, if you could have one treatment to add to your treatment tool bag that could affect every single one of these, why wouldn't you want to use it? And so as you go around this tree, I mean, looking at sleep apnea, there's somewhere upwards of 100 million people in this world dealing with sleep apnea as we speak. Alzheimer's dementia, certainly near and dear to me. There's no current cure for this. However, this is like a type 3 diabetes type picture. Dementia's happening, I think in America, every 65 seconds, someone's being diagnosed with it. Worldwide, that's every 3 seconds. And so if there's something we can do to the cerebrovascular glycocalyx to help lower the incidence of this, why wouldn't we, right? Moving all the way around, macular degeneration, heart attacks. I don't have to tell you, but you know this, the #1 killer of men and women is absolutely heart disease. Every 44 seconds, someone in this country has a heart attack. So it's not getting better. So something that we can use to help this would certainly be a great thing. Erectile dysfunction, right? 1 in 4 men around or under the age of 40 are now presenting with ED, right? That's not a sustainable problem. And I'd argue that erectile dysfunction is really a gateway, or at least an early sign that they have vessel disease somewhere else. One key thing to understand is an artery no matter where it is. So if you've got artery issues there, you're going to have artery issues everywhere. Kidneys, eyes, heart, liver, you name it. So again, moving all the way around, you guys get the rest of this stuff. But diabetes, in 20 years, 1 in 3 of us are going to have diabetes, also not sustainable. So we need to have something, another solution outside of what we already have to help sort of correct these issues. All right, so looking at the glycocalyx, as I said, there's that image again that you probably have imprinted in your head. It's this hairy sort of finger-like projection. What we know is it's very, very resilient. We can damage it through all those mechanisms I just mentioned, but we can restore it, and that's the key take home today. And that's how we do it. So here we go. This is arteriocele. I'm going to spend a lot of time talking about this. So this is first in its class. So the listener needs to understand this is not just one of many therapies proven to regenerate and restore the glycocalyx. This is the only thing that's been proven to protect it and restore it, literally the only thing. And so that's kind of why we're spending a lot of time on this, because it really is pretty revolutionary. I'll go through some historical, how this was discovered and all of that, but this is essentially what it is. It's a rare seaweed, contains these specialized sulfated polysaccharides used by the body to regenerate its own glycocalyx. Patents are pending on this. There's been a lot of money, a lot of sweat put into researching this over the last 20 years. And I'll go through some of the cellular, animal, and human studies as we go through. All right, so just kind of a brief story for you on how this all came to be. So this particular product in the mid-90s was found. There was a cardiovascular research team that was assigned with essentially just exploring ways to produce better outcomes in the cardiovascular space. And so they had different extracts of different things, and they were trying them on different patients for different outcomes. And one of the researchers came across, or a team of researchers came across this specific seaweed, or this sulfated polysaccharide, and they noticed that it was having certain effects on things, specifically things like blood pressure and erections, and in some cases, certain skin conditions. And they weren't sure what was causing this. And so, but they could tell that there was some clinical benefit from it. And so what that initial researcher did is assemble another team of about 9 to 10 more researchers. And it took them almost 10 years to realize what was actually happening with this particular extract is they were regenerating the glycocalyx. And so that's kind of how it all came to be. It was just part of a research team with several different compounds, and that's really how it was discovered. So when they discovered this and they realized how resilient it was, they needed to figure out, the researchers needed to figure out, how do we replicate this in a way that we can study it, right? We can't take a human glycocalyx and artery and do this in real time. We need to create an in vitro model where we can actually test certain compounds and replicate and try to get as close as we can to human anatomy and human arterial flow. And so that's exactly what they did. So they essentially created something that could quantify and show them a glycocalyx. It needed to replicate the pulsating flow of human blood through the artery, which is massively important to the health of glycocalyx. But the particular technology at that time had not been invented. So with the help, so Calroy, the makers of Arteriocell and the team, they're on the call, along with certain researchers at the Chinese Academy of Sciences, they developed this microfluidic chip. So this is now used for screening and studying what's called glycocalyx regenerating compounds, GRCs for short. So it's made of living human endothelial cells with endothelial glycocalyx, and you can rapidly test compounds. So this became very, very important because we needed to have data quickly to be able to research. So by using that fluidic chip, they were able to develop two key insights. The first one was it's very fragile. The glycocalyx in and of itself is very fragile and also resilient. And that Arteriocell itself, specifically the active compound of this sulfated polysaccharide, was very potent at restoring the endothelial glycocalyx. So then we're going to jump into this cellular study. So this was actually done using the microfluidic chip that I just showed you. And what they did is they took a normal glycocalyx, and we know, I've already told you that glucose or hyperglycemia erodes the glycocalyx in humans. And so they took a normal glycocalyx, and then they eroded the glycocalyx with glucose. And then they took that same high glucose state and then added different strengths, different dosages of Arteriocell, and you can see across the board. So normal, you have your control, and then you go into the groups that were given Arteriocell, and there was clear regeneration of that glycocalyx, even one dose up to 104%. So again, the conclusion is that it repaired and regenerated the damaged glycocalyx. For the functional medicine practitioners on here, as you all know, we go to the root cause of problems, we look at systems, and we look at a person as a whole and as one. And so Arteriocell, because it attacks or at least fixes something that's head to toe and is systemic, is a perfect tool to that entire functional medicine armamentarium. So looking at their research agenda, they have gone all in proverbially with this. They have put a lot of money behind this, and they're partnering with some pretty amazing clinicians across the country to get even more data that's powered and more scrutinized. Here's another study we're going to go through, leukocyte adhesion. So leukocyte adhesion, I sort of alluded to this earlier. It's the first step in atherosclerosis, but the other thing to understand is, I've already mentioned Alzheimer's. It's postulated that actually leukocyte adhesion is one of the first steps in developing Alzheimer's. So if that is in fact the case, it'd be nice to have something that could help this process too. So this was actually done with two groups of mice. This was the first group of mice. And what you'll see here, you have a baseline number of adherent white blood cells. Hyaluronidase was taken and removed the glycocalyx, all right? So that's our control. So when you remove the glycocalyx with that enzyme, you can see the adherent cells over double, and then they put saline inside of the mixture. And this is essentially the control slide, right? So 5 minutes, 15 minutes, 30 minutes, there wasn't really much done there. So the same amount of adherent cells stayed there. So what we're trying to do is find something obviously to lower the amount of leukocyte adhesion and the amount of cells. So then we had a second group of mice. Same thing. There's your baseline. We removed the glycocalyx with the hyaluronidase. And then instead, in this group, we took the 5 minute, 15 minute, 30 minute saline group, gave them saline, but also added the Arteriocell into it as well. And what you'll see here is leukocyte adhesion went down. So the amount of cells that were adhering went down. So the conclusion based upon this animal study was it prevented increase of leukocyte adhesion and inhibited the endothelium-mediated inflammation. This was another study done here locally where I'm at Baylor Hospital. They were looking at arterial elasticity. This was in 19 humans. And what they used is Max Pulse. For those that aren't familiar with that, it's an FDA-approved device that just measures plethysmography and arterial elasticity, among other things. And as you look across the board, the findings were an average increase of 89.6 in the arterial elasticity. You've got some outliers on here. We don't know exactly why that was. You can see human number 18 across the section here. We're not sure if he went outside for a cigarette or what happened there, but the study or the results still stand. There was that average increase across the board. Pretty impressive. Dr. Houston, a lot of you may be familiar with Dr. Houston. He's great. Love listening to Mark. Well, he actually at his Hypertension Institute took some patients and used Arteriocel on them. He took 10 uncontrolled hypertensive patients who are on max drug therapy. He put them on 1 twice a day of Arteriocel, and then he measured their blood pressure at 1 month, 2 month, and 3 month. And you can see the findings across the board here. Admittingly, yes, this was a smaller study, but the results are quite impressive. As you can see, systolic and diastolic. A lot of us on here that treat blood pressure know how hard it is sometimes to lower diastolic pressure. And you can see an almost an 11-point reduction there in 3 months for just twice a day of Arteriocel. But what's important to also draw your attention to, not just the systolic pressure dropping as well, it's those p-values. If you look from 1 month to 2 months to 3 months, the p-values increased, the p-values improved across the board, just showing more and more statistical significance. It's my hope, and I believe it's the goal, is Dr. Houston's actually going to do a bigger, more powered study, but this one was pretty impressive. Okay, going to go back to the discussion I had earlier about the lipid-rich necrotic core. We talked about calcified plaque. Now we're getting our heads back to this vulnerable lipid-rich necrotic core plaque that has the thin fibrous cap, the one that's really the major cause of coronary artery disease. This was done in humans. This was a proof of concept pilot study with 2 subjects, 1 man, 1 woman, both age 65. And they used MRI plaque view. Some of you may be familiar with this, but MRI plaque view is a very, very advanced technology. It's one that I wish we could order fairly routinely, but MRI plaque view itself is a new FDA cleared technology that differentiates between different plaque morphologies. So it can differentiate between lipid-rich necrotic core, calcified plaque, fibrous plaque. It kind of goes across the spectrum of plaque to help you understand what kind of plaque the patient has. But we studied these 2 patients specifically with Arteriocel, and this is what we found. So the study subjects ingested 2 capsules of Arteriocel twice a day for 60 days. And you can see what was found here. So again, age 65 across the board. What was found was, in that particular woman, a 64% reduction, net reduction in lipid-rich necrotic core, and then in the man, 46.8% reduction. That's pretty incredible. We need to take some time to talk about this because, you know, this is virtually unheard of. When you look at studies, like for example, when you look at statin studies, some of you may have read these studies before. There's a myriad of statin studies on what they do for lipid-rich necrotic core. And the great majority of them will show somewhere on the order of 1 to 7% reduction in lipid-rich necrotic core. Typically takes 2 to 3 years to actually get those types of findings. The best data we've ever seen on statins, one of them was out of the American Heart Journal in 2008. There was a reduction after 2 years of 25% of the lipid-rich necrotic core. And then in JACC in September 2011, there was one showing a 38% reduction in lipid-rich necrotic core, but that took 3 years. What we're talking about here is 30 or 60 days, so 2 months on Arteriocel twice a day, blew statin data out of the water. But the other thing to understand is the stenotic decrease. So we noticed that when we did the study, not only did it reduce lipid-rich necrotic core, but it significantly helped with stenotic activity. So it decreased stenosis within the vessel. And there's not one statin study on the planet that has ever shown that it can reduce stenosis as compared to this. So that was impressive. Now, this is the imaging findings from that particular human pilot study. It says 2 millimeters from the landmark. To the listener, the landmark is the bifurcation of the carotid artery. Okay, that's where we're starting, and we're going to start moving away. And I apologize, I hope on your screen you can see, but underneath lipid-rich necrotic core, it says maximal wall thickness. And as I said, you're going to see these next series of slides that decreases. Maximal wall thickness is now being considered a little bit more sensitive than the carotid intimal media thickness that some of us know from carotid ultrasound. So this one was pretty impressive. So you'll see the baseline on the left, and then after the arteriocele for 60 days on the right, you can see a reduction in the lipid-rich necrotic core, and you'd see a reduction in the maximal wall thickness. And again, I apologize if you can't see those numbers. As we move away from the landmark, 4 millimeters, you'll see again a reduction in the lipid-rich necrotic core and the maximal wall thickness 1.97 to 1.22. And then 6 millimeters away, lipid-rich necrotic core was gone, and the wall thickness also went down. So it not only reduced that nasty sort of stuff that leads to coronary artery disease, but it helped with, blood vessel, stenotic flow or arterial flow and reducing stenosis. And that's just not in literature that you'll find. Now, this is some new stuff that we added in, but I think it's important to see it. Carotid intimal media thickness. A lot of us might do this. There's some technology from a company called Vasolabs that I'll present here in a second. Some have coined this the mammogram of cardiovascular disease. I think this particular ultrasound has a big footprint in the country, and so a lot of us are familiar with it. But let's look at what arteriocele did for this. This is a 71-year-old female. The left is the before, the right is the after. And what was found when arteriocele was given twice a day for 3 months, there was a significant reduction in 4 out of the 5 plaques, and there was a reduction of vascular age by 1 year. Some of you may be familiar with these printouts, some of you may not. Next patient, 52-year-old lady, reduction of vascular age by 3 years, and there was a significant reduction of 1 of 2 plaques, and this was on arteriocele for 6 months. So again, left side's before, right side's after arteriocele. This one was pretty cool. 47-year-old man left before, again, right after, but there was a 6-year reduction in vascular age, and that had been on for arteriocele for 6 months. So that's a pretty impressive finding, especially when you look over and see, on the after page to the right, it has all the ones that this gentleman did: March 2016, November 2017, September 18, May 2019, and the percentiles. So you can see the arterial age is just going down just by the intervention of arteriocele. So impressive. Okay, so just as a summary of these findings to date, we know that arteriocele repairs and regenerates the glycocalyx. We've proven that in cellular, animal, and human studies. Inhibits endothelium-mediated inflammation, improves endothelial function and arterial elasticity, helps maintain normal blood pressure, and it might stop and reverse these atherosclerotic plaque developments. The research agenda for this year is pretty aggressive. It's exciting stuff. So some of these have been approved pending initiation. So randomized hypertension study, atherosclerotic plaque study, and ApoE knockout mice. And then there's some additional human studies in design. Atherosclerotic plaque without statins, plaque with statins, and then diabetic neuropathy pain and erectile function. So these things that are coming down the pipe. So stay tuned, because we'll present the data once we get it. So use in clinical practice, that number's actually gone up. There's more physicians now than that, but there's a lot of physicians now using this. Lots of patients have been given this. We're talking about over 3 years of data. There's no known contraindications or adverse side effects. I use this routinely in clinical practice, and I'm very happy with the results. And I can attest to that. There's been no adverse events in any case that I've seen or heard of. And there's been case reports of atherosclerotic plaque regression, lowered high blood pressure, improved erectile function. I mean, I've seen inflammatory markers go down. I've seen certain plaque markers go down. It's been pretty impressive. So who should take Arteriocel? I think the listener is probably asking who shouldn't take this, and that's probably what I'd ask too. But definitively, all men and women age 45 or over for prevention. All men and women, regardless of age, diagnosed or with familial risk of cardiovascular disease, stroke, high blood pressure, atherosclerotic plaque, diabetes, diabetic neuropathy, ED, and other circulatory issues. Incidentally, you can go up to 3 times a day if you need to for diabetic neuropathy. There's been, for the pain associated with that, there seems to be some good response there. Here's some pricing of the product. You can talk to some of the representatives from Calroy about that if you're interested. And these are some of the ways that you can get Arteriocel. What's important for you to know is this is not direct to consumer. This is only for us as clinicians and prescribers to carry in our practices. So it's not something they're going to go find in a local store or online. This is through prescribers and prescriber accounts. So takeaways. Some of this I've kind of hit, but really I want you to consider the next time you think about an intervention to lower an inflammatory particle in the vascular lumen, or lower toxic burden, or lower oxidized LDL, start thinking to yourself, what does this do to the glycocalyx? Some of you, as I said, may know the glycocalyx. Some of you are just now learning about the glycocalyx, but it's massively important to ask yourself, and then anybody that comes in, a vendor, a rep from either a pharmaceutical company, a nutraceutical company, whoever it is, what do you know about the glycocalyx? What does your product do to that? And I can assure you that I've been doing that myself to people that come through my offices, because it's just a very, very important structure. We've got to pay attention to it. Cardiovascular disease is the #1 killer. There's a shift away from the lumen into the endothelium, which is the way it should be. Not discounting the lumen, but more focus in the endothelium. There's a lot of research around the glycocalyx. And as I've said, it's a resilient structure and we can regenerate it pretty nicely. And the glycocalyx and the endothelium work together. You can't have one without the other. They're important to each other. Arteriocel itself is the first in its class. It's the only thing proven to rebuild the glycocalyx. Lots of research, lots of money has been put into that. And I've mentioned how it was derived and really what it consists of. And I've gone through the human studies for you. The safety data is pretty strong. There are no known contraindications. And I've already mentioned it's sold through licensed healthcare professionals. Now, just because of the times we're in, I'm going to hit real quickly some COVID slides here. This is just giving you just a general overview based upon the CDC and really up to date on some of the spectrum of infection that we're seeing and the spectrum of illness. And so a great majority are mild cases. There's really no severe symptoms there. The severe cases really involve obviously lung involvement, hypoxia, 15% of those cases, and then some of those have matriculated into critical disease with multi-organ failure, respiratory failure, shock, and then the majority, or at least several of the fatalities have really involved respiratory failure, septic shock, or as I said, complete organ failure. So we know that ARDS or ARDS is a major complication with patients with severe disease. Other complications that have been seen so far are arrhythmias, acute cardiac injury, and myopathy, as well as shock. And then a lot of the majority of the case fatalities in China occurred in patients with advanced age or these particular comorbidities. So, you know, cardiovascular disease, I think we've all heard, is one of the major risk factors of you know, having a worse outcome with COVID. And so, as you all know, cardiovascular disease is not something we treat with one modality. We have to surround it and help prevent it and help treat it. So there's several ways of doing that. So looking specifically at COVID and the endothelial glycocalyx, we already know, okay, we didn't have to learn this tonight. The literature's been out there that the glycocalyx is massively important to cardiovascular health, endothelial health, and all of that's really the foundation of it. And so when you look at COVID and what the illness is doing, you know, these ACE2 receptors are scattered throughout the body. A lot of them are concentrated really in the pneumocytes and the enterocytes of the gut, but they also include the endothelium and the cardiac cells. And so those have to be paid attention to as well. And the vascular system itself has been implicated in some of the infections from COVID, but the inflammatory damage, which again is in those pneumocytes. And we know that when pneumocytes are damaged and lugs are inflamed, the endothelial glycocalyx essentially goes away, which ultimately leads to all sorts of fluid shifts and fluid overload, changes coagulation factors through the liver. Patients become hypercoagulable, and it just leads to this cascading sort of dire storm that they're going through. So maintaining good cardiovascular health is obviously certainly important. COVID, can progress to these sort of injuries I've discussed. Septic shock and fluid management I've also discussed. The glycocalyx is massively important in sepsis, cardiogenic shock, any of those things. A healthy glycocalyx defends against endothelial damage and sepsis. And preserving the glycocalyx would protect these microvessels from fluid leakage. That's in references I'll provide here in a second. So, and fluid resuscitation is sometimes difficult, especially if the glycocalyx is compromised or otherwise damaged. So, there's no leap we're making here with arteriocele and COVID and glycocalyx. What we're simply doing is creating a sense of awareness about the current literature around septic shock, pulmonary edema, ARDS, and the glycocalyx essentially being gone. And in dire times, sometimes certain measures have to be taken to help replete that and repair that. And so that's really what it is. It's just more about you being aware of something that can regenerate the glycocalyx, something that what happens to the glycocalyx in systemic injury, and really just understanding that physiology, and just having an awareness of COVID-19 and what it seems to be attacking right now. Those are the references for that. Here are the references for arteriocele, and here are some ways for you guys to contact Calroy and learn more about arteriocele if you're interested in finding out more about it. I think we're at the point now. I think I'm right on my time limit. We're going to probably open it up for question and answers if there are any.
Thank you, Dr. Rice. Super informative. Really appreciate your time tonight and appreciate all the people that have joined in. This will be available through replay. We will be sending it out. We had a lot of people tell us that just due to circumstances, they couldn't attend live and they wanted to have it available for replay, so we are doing that. Got a couple questions that have come in on the open line as well as some that have come in privately. Here's a couple questions, and Dr. Rice, I'm going to open these up to both you and Dr. Chen, and you guys can figure out what to do. But is arteriocele used daily, or is there an on-off strategy for long-term use?
Yeah, I can field this one. So one thing that you have to understand about arteriocele is that it's really uncharted territory that you have not only a preventive, but you have a therapeutic. And so it's my opinion as a clinician, if you want to continue to get the gains from regenerating the glycocalyx, you're not going to stop the modality. And so I'm not aware of any ramping up phase and then lower, because with some products, as some of the people on the call know, you can sort of load a patient with high dose and then back off to a maintenance dose. With this particular product, I don't know that it's been researched that way. You guys can chime in if it has.
Yeah, we do have some practitioners that use technologies to measure arterial health. For example, that max pulse technology you talked about, that once they help them receive a arterial score of 3 or higher, with 1 being the highest on that scale, they will many times put them down to 1 capsule a day instead of 1 BID. And as long as they maintain that score, then they can keep it that. So there are some patients that have been able to successfully go down to 1 cap a day, which effectively cuts their cost of product in half. So,
sure.
Next question here. What about its blood thinning effects? Can someone take this if they're on blood thinners?
Absolutely. Yeah, there's no known problem with arteriocele and blood thinners or hypercoagulability or any of those sort of issues.
Okay. Another question, could this product potentially help someone with arterial fibrillation?
Yeah, so the interesting thing is if you can restore endothelial health through the glycocalyx, you're restoring the function of superoxide dismutase. So you're going to get better nitric oxide production. better nitric oxide production leads to better vascular health. And it really becomes, it helps with contractility and, it's sort of an inotrope. So I think if you have ongoing glycocalyx health through restoring it and regenerating it, there's no reason it shouldn't help any sort of underlying, cardiac pathology. Specifically with arrhythmias, I haven't used it, for fib or any of the other ones out there, but it stands to reason that, the glycocalyx itself, if you can keep it restored and you're not having a compromised, ability to affect what goes in and what goes out, then it should only help. It's certainly not going to worsen any outcome with an arrhythmia.
So thank you. You've already answered about the contraindications. How does arteriosol deal with oxidative stress and inflammation?
How does it deal with it? Okay, so as I said, the main regulator in our body is that enzyme. As I said, it's that internal pharmacy. It's that superoxide dismutase, and that's inside of the glycocalyx, right? So that's stuffed inside of that furry coat. And so if that's pumping out nitric oxide, superoxide dismutase is going to block and catalyze superoxide anion. And doing all of that is absolutely going to lower oxidative stress. So it's going to fix any of that internal rusting of those particular pipes. And inflammation itself, it just helps through all of the proteins itself. And just once you have endothelial health and restored glycocalyx health, you're not going to have inflammation. Is really when the single layer endothelium, the vascular endothelium, is compromised, and you start having kind of that plaque buildup on the other side that could then explode. But if you have an intact glycocalyx, you're not going to get to the point where any inflammation is going to really affect that or even cause any of that stuff, as long as you can keep that furry coat intact and not compromised.
Great. I'm going to direct this question to Dr. Chen because I know that he's been doing a lot of research in this area. Is there any interaction with ACE or ARBs?
An interaction with ACE and ARBs of arteriosol?
The question just says any interaction with ACE and ARB, so I'm guessing that it means with the arteriosol or with the endothelial glycocalyx. What's its role?
I mean, ACE and ARBs are obviously involved with endothelial health and regulation of blood pressure, and the glycocalyx. Endothelial glycocalyx in a way have the same function, although maybe through different mechanisms. So I'm not aware of any direct research related to endothelial glycocalyx with ACE and ARBs, but if you look at the impact of the endothelial glycocalyx on the endothelial function, and especially related to the pressure control, there are certainly a possible links between those, or there could be some indirect effect of the endothelial health on the ACE, because they are expressed on the surface of the endothelial cells, which is underneath the endothelial glycocalyx. I don't know whether that answers the question or not.
Yeah, the person that asked the question said yes, so thank you. Here's a question. In general, is seaweed consumption associated with reduced cardiovascular disease? And that might relate to if this is unique to this seaweed, or if in general, any seaweed.
I am not aware of ongoing seaweed consumption outside of its iodine, selenium, and micronutrient compounds ultimately helping. I'm not familiar enough. I know that I've had this question before about, can I just eat seaweed? Well, no, this is a specific, type of seaweed Sure, eat seaweed for its own health benefits, but I don't think you can make the same claims that arteriosol can make.
Thank you. Question, and Dr. Chen, this is probably good for you because you were involved with this study, but have you done any testing using Itamar's Endopat for endothelial function before and after use of arteriosol?
Very early on, we used Endopat in one of the human studies, and we specifically look at the acute effect of arteriosol on some healthy human subject after they consuming a high sugar, high fat meal. And in that particular study, we see a response of Endopath, specifically RHI, reactive hyperemia index, to the arteriosol. And we monitored the RHI for 8 hours after the consumption of the meal, and arteriosol prevent the decrease of the endothelial function in that particular study. Yes, we did some study right now.
Okay, thank you. Are there any contraindications or problematic interactions with any other drugs, nutrients, or herbs?
None that we are aware of.
Thank you. Question, what about the hydrogen peroxide produced from SOD? That can be metabolized to hydroxyl free radicals, which are even more damaging. How is it affecting catalase?
How is it affecting catalase?
Yes.
Dr. Chen, you may answer that.
Well, catalase is an antioxidant enzyme that would prevent this from happening. So basically, you need to have a strong antioxidant system inside your body, which is the antioxidative enzyme catalase. So there are different ways of doing that, but basically, yes, you need to keep a strong antioxidative capacity of the body. But from the glycocalyx perspective, the importance of endothelial glycocalyx is really related to SOD in this case, which is prevent the superoxide ions turn into peroxyl nitrate by reacting, in this case, actually with nitric oxide. So this is a very complex system in our body. You need to have a balanced system to put everything in check, so you don't overproduce one thing over the other and end up with some undesirable result.
Thank you. And we've got time for maybe one more question. Questions that we didn't get time to today. We do have your email address from your login, so we will get back and answer those questions. And I was asked a couple questions privately along these lines, which is about how long does it take for people to, do you see clinical effect? This question in particular said, how long will it take to help people with peripheral vascular issues such as cold hands, cold feet?
Yeah, that's a great question. And if I was on the other side of this, I'd be wondering the exact same thing. And it's a really hard question to answer because of the uniqueness and the bio-individuality of all of these people we're seeing. So I can try to give you my experience the best way I can. I don't know that you're going to get an answer across the board that is going to satisfy, you. But ultimately, if I've got a metabolically deranged patient with known peripheral vascular disease, coronary artery disease, high inflammation, objective evidence of there being stenotic plaque, or if I have the technology to show me vulnerable or rupturable plaque, in my experience, it's about 3 to 6 months out is when I would expect to see some change. The problem is, as you can certainly understand, if you're a prescriber, you're not going to do one intervention. If we were left to our devices to just say, you can only have one thing, do it for 6 months, maybe so. But all of us are working on nutrition, gut health, hormone health, thyroid health, sleep, stress, while layering in, like me, these types of things if I see objective evidence. But I can give you kind of a range that if the one symptom you're looking at for peripheral vascular disease is peripheral temperature not being cold, if you're going to see a response, I would imagine you would within 6 months conservatively. But what I would tell you is if they're consistently using arteriocel, look at two things. One, erections, if it's a man. Two, blood pressure, because those things we know that nitric oxide is going up. So those are two things we can quantify. It's like when I'm treating a patient with known plaque. They don't feel it, they don't know it. They just have to wait till an event happens, then they feel it, then they know it. So I said, you're not going to feel your plaque going away, but you're going to notice if your erections are better or your blood pressure is getting better. Those are two things they can kind of quantify. Other than that, they're putting their faith in you to do that whatever that diagnostic is a few months out. But 3 to 6 months is usually what I do to repeat whatever it is that I'm looking at.
Well, thank you. And thank you to everybody for being on the call tonight, Dr. Chen and Dr. Rice, for your contribution. I very much appreciate it. For all of you listening, especially taking time during these extremely unique times to learn about this and the technology behind it. If you'd like to register for a practitioner account or get more information, you can go to arteriocel.com. You see it on your screen now, or you can reach out to us through our support email or our 1-800 number. As I mentioned, we will answer the rest of the balance of the questions that we weren't able to get to. And thank you very much.